A warm greetings to all researchers, scientists, professors, and students. On behalf of ICWR, I welcome you all. International Consortium of Water Researchers is a non-profit consortium, operates with an objective to connect water-based research and researchers and scientists all over the world. It is promoted and supported by international and national advisory members of various research and educational institutions. ICWR operates with the interest towards enriching knowledge at 4 different stages as water fellows, water experts, young professionals, and student members. ICWR does this through organizing webinar series, training and workshops, and on water-based modeling and annual virtual conferences. We also further our objective by operating effectively among the water-based research aspirants at all levels. So far, ICWR have organized 3 webinars through international experts like Professor Eric van Helbusch from University of Paris, followed by Professor Ali Alte from University of Sydney, Australia, and Professor Esther van Herden from iWater Private Limited, South Africa. With this short introduction about ICWR, I would like to introduce Professor Frederic Coulon from Cranfield University for the 4th webinar of ICWR. I feel very happy to introduce him to the community of water researchers. Professor Frederic Coulon is having more than 20 years of research experience in environmental science. He is having 180 plus research articles, books, and book chapters. His area of expertise is environment and science and health, soil resources, waste management and resource efficiency, and water science and engineering. He is having more than 3,600 plus citations, and his research activities is mainly focused on activities and interactions between the microbes in bioengineered processes. He is into the crude oil and refined products biodegradation, the diversity and roles and functions of various microorganisms in polluted environment he is working on. And he is trying to study about the different microbial adaptations and responses to the recalcitrants in the cold environment as well as in the different kind of pollutants. He is also into the environment and human risk assessment, diagnostic strategies. I think the diagnostic strategy and risk is into the environmental fate transport and exposure modeling. And he's working with a lot of emerging contaminants and hazardous residues. And I feel the entire Cranfield University is working together on water-based research. And I feel very happy to introduce him to the community of water researchers. Professor Frederic, now the platform is yours.
Thank you very much, Panaman, and good evening to everyone, and thank you for joining us for this webinar. I'm very pleased to be contributing to the initiative. I find very excellent. So today I'm going to share with you work we've been doing on engineered nanoparticles and what are the potential impact on wastewater treatment plants, what the fates, the transport, and whether is a concern or not. This work was carried out by one of my PhD students, and we had also a postdoc involved. So we were funded by the transatlantic initiative between the US and UK, the TINY project, and also by the Commonwealth Scholarship supporting the work. So what did we look into nanoparticle and specifically more engineered nanoparticles? It's because they are on our daily life more and more used across a various range of products, also clothes, sport equipment, shoes. It can be very useful for anti-odors, all on the spray you might use on your body as antiperspirants, but also in various range of product for conservation. And so far, those have been the most reported is a zinc oxide, titanium dioxide, and a silver nanoparticles. So we dedicated our attention to these 3 engineered nanoparticle in wastewater treatment plants. And despite it might look quite dilute in the system, and you do end up by having concentration happening in our wastewater treatment plants. And that is where we get all going through the wastewater. And when they get concentrated, we want to understand where they do end up and whether they will affect the process. And in UK, in Europe, but also more widely, is getting more and more, there is interest to reuse the biosolids for anaerobic digestion and produce biogas. So in these projects, we were also interested not only on the freight transport during wastewater treatment plants, but also the impacts, potential impacts during anaerobic digestion. And obviously, to close the loop, when you got the digestate, and then you might apply it to land as a soil amendment, whether there is any potential risk. And also, you might have some liquor or leaching, so looking at the quality of the surface water. So if you look in terms of the range of products where you got engineered nanoparticles being used for the product design and application. Here, just an illustration, you can see pretty much over the last 15 years, the range of products using or having engineered nanoparticle keep increasing almost every year. In terms of concentration, you might, there is quite a wide range published in the literature. But for zinc oxide could range anything between 12 to 25 milligram per kilogram of biosolids, silver 7 to 39 milligram per kilogram, and titanium between 3 to 100 milligram per kilogram of biosolids. So our key interest here was really to understand from all this process what happens during our wastewater treatment plant processes, and whether any effluent might be posing potentially environmental issues for river stream. But we were also willing to understand what happens during the wastewater treatment plant. So do we got agglomeration? Do we got aggregation within the solids? Is there any reactivity being induced by the presence of the engineered particles? Are they bioavailable, or do we got some internalization issues? And if they are bioavailable, would they bioaccumulate? And the problem with the bioaccumulation is once you got a release in environment and potentially have posing an environmental issues. Would they affect sorptions? So we know the nanoparticles do not behave as their metal counterparts. And that is very important to take into account. So we're really interested to understand how these nano particles behave during the wastewater treatment plants, and what are the, if you want, the chemistry mechanisms, but also the potential effect on the biology of our wastewater treatment plant. So when you look at the typical wastewater treatment plant, you do have two phases. You got the aerobic phase, where you mainly have the activated sludge. So you are effectively injecting, aerating the water to encourage the degradation of the solids. And we do have also the anaerobic digester, where we could use the sludge for further digestion, having energy recovery opportunity, nutrient recovery, and obviously having opportunity to have fertilizer. From the literature, it's been well published and acknowledged the nano could have potential effect on the various steps, biological processes occurring during the treatment processes. So we were particularly interested to understand whether engineered nanoparticles will affect the nitrogen cycle. So the nitrification, ammonifications, and nitrates. And also we were interested to understand whether the nano will affect the anaerobic digestion, so all about the fermentation, the acidogenesis, but also around the methanogenesis. We are really lucky in Cranfield University. So if you haven't heard about Cranfield University, we are about 40 minutes up north London, or maybe Luton Airport, if that talk to you. But we are a former RAF base. So we are a military base formerly, and that means we do have a real wastewater treatment plant at the university, which our PhD student researcher can use. So here you can see we do have a typical trickling filter process. We've got some aeration tank and tertiary treatment. And you can see these two buildings where we got our pilot hall. That is giving us about 400 square meters of facility to do pilot testing research. And it's where we've been using real effluent from wastewater treatment, and we have been testing the impact.
Hello, Frederic. Can you hear me?
Yeah.
Frederic, some participants are saying voice is not audible. Can you increase the volume in your computer?
Yes.
Yeah.
Here is the max now. Is it
yeah, thank
better?
you. Yeah, it's fine.
So in the pilot hall, we did set up 3 pilot scale treatment plants. One was a control as it is, so the sludge from the real wastewater treatment as it is. And then we have a line where we spike with the metal counterparts for silver, titanium, and zinc. And the third line was spiked with the nanoparticulates. So silver oxide, titanium dioxide, and zinc oxide. The settings of our pilots, so you had the real wastewater treatment plant we use as effluent, which was reaching the primary clarifier, 180 liters, and that was to feed the activated sludge with 300 liters. And we were spiking at the activated sludge entry. So here you can see the concentration for silver, titanium, and zinc. So we were using a rate of loading of 14 ml per minute to feed and to spike our activated sludge. Typically, from an activated sludge, you will get the secondary clarifier, and then you got your effluents, but we are collecting the waste activated sludge, so the solid, which is second, and that could be recirculated in activated sludge, or if it's reduced enough, we could move to a blending tank before feeding the anaerobic digester, so where we got the primary sludge with the second waste activated sludge. We had a second entry as well for the spiking, so the spiking from the effluents in the activated sludge, but also spiking looking at the primary sludge itself, and if you were going straight away for anaerobic digestion. So we were interested to understand the partitioning from the liquid phase, but also with the solids. Then you have the digestate, and then going toward the holding tank for the dewatering and getting the digestate cake and filtrates. Our targets when we choose the spiking concentration was to achieve 250 milligram per liter of silver, 2000 milligram per liter of titanium, and 2800 zinc milligram per liter. These concentrations were the worst case scenario we were willing to look into based with our discussion of our partner in US. In terms of the operating settings, we had the settled wastewater was at 45 liters per hour. The return activated sludge was at 22.5 liters per hour. We had an hydraulic retention time of the process of 8 hours and the sludge, activated sludge age of 10 days. For the anaerobic digestion, we have 15 days. So we run these pilots for 3 hydraulic retention time to allow for acclimatation before to start the spiking. So here is an illustration how it looks like. So you can see the 3 lines with the tanks. So where you got a line spike with nanoparticle, the control, so it's just the sludge as it is, and then the metal spike line. You might argue, so when you are dealing with such large volume, it's really challenging to get replicate or triplicate. It's not like going in your lab and doing the bench scale. You must realize you are about 900 liters of water, and you are doing some treatment where you're spiking with heavy metals or spiking with nanoparticles. So effectively, we had some risk assessment in place, and also with the nanoparticles, so you do have some limitation, and you have also to engage actively with the regulator, because you cannot release simply your effluents in the environment. On the back, you can see the primary clarifier. So that is fed directly with the water from the wastewater treatment plants. And here you got the anaerobic digestion cell, and then the other part you got the clarifier. So if we zoom a bit, you can see better the settings. And for the anaerobic digester, we had also a heating jacket, so that was allowing us to have control temperature over the process. And the primary and secondary also could be selected or shut off as needed. Here you see the filter press. So when after the digestions, and then you could see the sludge cake we were getting out from the anaerobic digestion process. So that is our setup and the parameter we've been looking into. So the first one is the microbiological parameter. So looking at a typical indicator of biological activities, respirometry, total abundance. We've been using phospholipid analysis and phospholipid ether lipids, so more for the archaeal and methanogen specifically. We have done also some macrotox assay and use scanning electron microscopy to look at potential damage on cell membrane during the process. These were combined with physicochemical parameter. So again, to assess whether the entry of engineered nanoparticle would influence the process. So if we start to focus first on the activated sludge phase, so you remember we do spike at the activated sludge stage, and we have also the primary sludge. So we've been looking at what would be the fate of the nanoparticle silver. So in that case, we've been looking, you got the control, so the pilots with the wastewater as it is, the metal salts. So here is the sludge from the wastewater, which we spike with the metal salts. And here is the nanoparticles, so is the wastewater we spike with silver nano. And clearly, if you, so from the control, 100% is pretty much in a waste activated sludge. where if you do spike with metal and nanoparticle, so in other words, you increase the concentration, almost 90% will be an activated sludge, but you can see there is a small fraction that might be recirculating in the water phase, so going back to the activated sludge. When you look at titanium, again, the control clearly all goes to the waste activated sludge. And even on the spike, so you are really exceeding concentration, you will find naturally almost all of it would still go to the waste activated sludge. So little is being resuspended and going to the liquid phase. Sorry. And the zinc, again, the control, all of it go to the waste activated sludge, where the metal salt and nano, we do have a small proportion being resuspended. So if we try to sum up these, so you had the control, the metal salt and nanoparticle. So in the control, these three metals are present as background from the wastewater. In this metal salt is where we did spike, so we add extra on the metal salt, and the nano is where we spike with the nanoparticulate form of the silver, titanium, and zinc oxide. So the good news is nothing goes to the final effluent. So that means your metals or nanoparticles will mainly partition in the activated sludge. A normal concentration all go to the waste sludge, so they follow the solids, where if you start to add extra, so you are over, then you do have a small percentage, about up to 10% for the metal and about 3% on the nano, that might be dissolved and therefore being recycled in the activated sludge stage. So in terms of these, bearing in mind you are in your activated sludge, we look at the biochemical oxygen demand. And if you compare the control to when you spike with the metal salt or the nanoparticulates, you can see the difference are not very significant. There is no significant difference, sorry. So in terms of biological activity, even the excess of metal or nano didn't have much influence on the biological activities. In terms of phosphorus removal, we didn't see much significant difference either. And the only difference we've been observing was for ammonia removal, where clearly the metals are having a negative effects or inhibiting ammonia removal. But if you look at the nanoparticles compared to the control again, we didn't see any significant difference. In terms of aggregation within the activated sludge, so you could look at the floc size distribution. So in the red, you got in presence of engineered nanoparticles, and blue and green is where you got the control and the metals. So looking at the effects, again, only the nanoparticle were effectively influencing the formation of large floc. And you can clearly see the control and metals were able to go up to thousands, where with the ENP, we didn't manage to get these large distributions. So potentially suggesting the nano are affecting the EPS productions. In terms of whether the nano or the metal salts influence the activated sludge volume index, well, again, here we didn't see much effects except within the first 13 days. So once you need to remember, before that, we had a 3 retention time to allow for to have a stable process. and just after here, you start the spike. So when we had the metal at the first parts, we also noticed there was some excessive foaming issues, which possibly are due to filamentous organisms. But we couldn't find a direct correlation on whether metals were stimulating or filamentous organisms. If we look over the 230 days we've been running the pilots, in terms of the pH, we couldn't see much difference between control, metal salts, and engineered nanoparticles. The BOD were same. The only difference, as we mentioned earlier, was for ammonia. And we also looked at the TSS, so back to the floc particles. But overall, we couldn't find much influence on the activated sludge. In terms of microbial abundance, we do have a log lower compared to the control, but the difference were not significant. But what we could observe when we've been looking at the biomass using phospholipid analysis is we couldn't find any biomarker for ammonia nitrate oxidizing bacteria. So that might indicate they can be sensitive to high load concentrations. We could also identify firmicutes, and they were associated with the phosphorus removal and sometimes being negatively affected with the concentrations. We've been looking at whether the nano had any effects on bacterial cell wall. So here you got is from the activated sludge, and you can see clearly here some damaged cells with the presence of the nanoparticles. So potentially indicating also lipid peroxidations. We had also observation of aggregation or agglomerations. So that could be also affect bacterial activities and also be releasing ions that could potentially either enhance or attenuate the nanoparticle effects on the microorganisms. If you look in terms of activity and the specific oxygen uptake, you can see the metals were effectively stimulating somehow due to potential stress, the oxygen uptake rates. And we could also notice with the nanoparticles, but again, if you do compare with the control, the difference were not significant. In terms of how the community evolved within the activated sludge, so we did monitor from T0 after 30 days, 90 days, and 150 days. So here we did parasequencing, so NGS amplification, and looking at whether we could see any shifts or change in the community. And here you got the control in this left-hand side, the metal salt, so the line spiked with metal and the line spiked with nanoparticles. And clearly you can see some change in proportion of the key member of the community. So we can see some shift in diversity and richness in the activated sludge, where you tend to reduce the richness when we its fact with metal or nanoparticles. So effectively, the activated sludge, overall, you didn't find significant effects on the biological process itself. Clearly, the nano and salts tend to partition to the waste activated sludge. And that if you get mixed with the primary sludge, so then you reach the anaerobic digester. So now we are going to focus on the anaerobic digestion and looking also on the filtrates and the digested cake, so the end products. So for the nanosilver in the AD process, in the control, all the background silver is in a cake. But when we did look at the metal salt spike line, you can see there is a fraction in a cake and a fraction will be still in the liquor phase. So when you press the digestate at the end, you remember you got your filter press, you got your cake, so you do have the liquor. So here is the fraction that would be effectively in a liquid phase, where here is a fraction in the digested biosolids. And the nanoparticle, we find a similar trend. So effectively, this is something to watch out. Same issues with titanium. So in the control, in that case, where you could see a small fraction being dissolved and being present in the liquor. And for the metal salt and the nanoparticle, again, most of the titanium is in the solids, but you do have a fraction that is in a liquid solid. And similar issues with zinc. So if we're trying to summarize here, if you do have your control, so that is background concentration, you will find in wastewater, most of the metal salt would be in the cake. And for silver, it was 100%, for titanium, 99.9%, so you got about 0.1%. But zinc is much more mobile, so you're very likely zinc will be redissolved and be in the liquor as well as in the cake. If you now look for the metal and nanoparticle spike line, you can see there is in the cake base, there is a non-negligible fraction that would be in a liquor. So roughly speaking, you got between 30% to 70%. So most of them will partition in a cake, but you might have also resuspension in a liquor. And that is something to bear in mind when you do operate wastewater treatment plant and you have anaerobic digestion and you have your digestates, which you might dry out. And so you might have some trace of the either nanoparticulate or metal salts. And you can see, hello? And you can see silver in particular might be resuspended. For whether, so the next point was, do metals or nanoparticles have effects on methane or H2S productions? So if you look here, the control on the white dots, the black one is the nanoparticles and the gray dots is the metal salts. So we had a quite lag phase for starting compared to the control in terms of methane. But after almost 60 days, then the methane production start to peak even in presence of nanoparticles. And when we look at this, that was also associated with a lower H2S production. So in that case, it seems that the presence of salt, metal, or nanoparticles had some beneficial effects by reducing H2S production and therefore encouraging methane productions. So here we go a bit a wider zoom on that methane productions. And we had some spike up and down. So you could see here a good gas production and then getting in a phase where decreasing and take time to recover. And it was the same for the metals and the nano. And that is probably also one issue probably to the washout of the biomass, because we have obviously to feed back the pilot plants on a regular basis. So that's something we is potential also influencing the gas productions. Looking on this gas production, so obviously before methane is all linked with the volatile fatty acid productions. And one thing we notice is you do have co-accumulation of acetic acids and propionic acids, especially when you got the metal salts. So the gray bar and the dark square is the nanoparticles. So there is something to watch out, because obviously if you are accumulating acetic acid, then you might end up by shifting from methane production to hydrogen production. We did also some separate tests. So we did BMP tests for the biomethane potential tests. So here you can see the setup we have at Cranfield, and we did a range of concentrations. So from concentration you might expect within wastewater treatment plant up to worst case scenario. And clearly you can see the worst case scenario will have quite a significant effects on methane production with almost inhibitions with the metal counterpart. Looking at the biomass dynamic within the anaerobic digester, so we use phospholipid acid for the bacteria and phospholipid ether lipids for the archaea. And you could see the abundance were quite different if metals were present compared to the nano or compared to the control. So within the reactor, we could see some difference in shifts and abundance of microbial groups. Looking further on these details is clearly the firmicutes, Proteobacteria and bacteroidetes were the most influenced by the presence of nanoparticle compared to when we had the metal or just the control. So here you got the control on the left-hand side, the metal spike at the middle, and the nano spike AD in the right-hand side. So looking in details, the firmicutes were 2 times slower when, so the green are for the firmicute, 2 times slower when we had the nanoparticles. We had also the Proteobacteria and bacteroidetes. They were 3 times slower in the nano. But in contrast, we had Fusobacteria and Actinobacteria were 8 to 2 times higher in the nano spike digester. So that highlighted here with the blue. We were quite interested to have a bit more insights for the methanogenic community. So we've been looking specifically for the phospholipid ether lipid marker and the E20 and E21 in particular. And so clearly you'll see the biomass as an indication based on the phospholipid ether. is one order magnitude lower. But what we were very interested to see is clearly a shift over time. And at 130 days, we did see some specific cluster appearing, especially when we had the nanoparticles. So we did further sequencing and find that this was mainly due with methanosarcina. So you got the control digestate, the spike digestate, and the nanospike digestate. And you can clearly see when we had in the nano spike digestates, all these key methanogens were not present, but we had methanosarcina, which was the dominating methanogens. And that was really suggesting you do have adaptation and methanogens that can tolerate high concentration of nanoparticles. So we find some nanotolerant species in our study. In terms of inhibitions, either by the presence of the nano or metal in the digester, so we use microtox, where you got your luminescent tests, and we have a varying acute toxicity response in presence of the nanoparticle or the metals. So bear in mind is EC50, so the effective concentration that will inhibit by 50% the luminescence. So the lower is the value, the higher is the toxicity of the metals or the nano. So we had in some place, you could even see the metals were having a higher inhibition effects compared to the nano. But if really we look over the time, so you were the onset 20 days, 40 days, 120, 140, 50 and 70, there is no really major impact on the process. So if we're trying to sum up, well, the nano effect on the activated sludge, we couldn't really find inhibitory effect on the biological removal process, so the efficiency was not really lowered. You do have some similar stimulatory effects on the specific oxygen uptake, and that probably due to stress, so the bugs are able to respond to it. Little effect on the influence of the nano for the sludge suitability, so that was mainly with the floc size distribution. The bacterial abundance was reduced, but the activity was still present. We did see some cell wall damage due to the presence of the nano, but we found bacteria can be resilient, and especially acidophorus and rhodophorax in activated sludge were the most dominant and resilient. There were some effect, however, on the nitrospira and ternary cuts, so linked with ammonia reductions, but we couldn't really see any significant inhibitions or effect on the overall process. So you do have a shift in the community, but the community adapt and become resilient to the stress. For the anaerobic digestion, we've seen a reduction in production of H2S. So that means it was associated with a reduced abundance of sulfatoreductor bacteria, and therefore that was positive effect for the anaerobic digestion performance because we had a higher methane productions. The nano can be bioavailable in a filtrate. So after digestion, when you do filter your cake, you will get between 26 to 59% possibly of your nano being still in the liquid phase. The firmicutes were dominating, but for the bacteria, But for the methanogens, Methanosarcina was identified as a nanotolerant species. So overall, the majority of our dose material is as a salt, metal salt, so dissolve, or as a nanoparticle, so engineered nanoparticle, is really much associated with the solid component of the wastewater. So that is quite good news for aquatic system, and is also for the transport of the nanomaterial, is very likely most of it or a large fraction of it will end up with the biosolid fractions. There is evidence or some evidence of damage to bacterial cells, but it doesn't contribute to an underperforming biological processes. There is also some stressing happening on the biota, but is also stimulating in the same time response of the biological component of our wastewater treatment system. So overall, the treatment system within the settings we had has been quite resilient. We did have resilient to the increased dosage tested, and bearing in mind the level were considered as very worst case scenario. And so we couldn't find until now significant differentiation on whether you got metal salts or the nanoparticulate form. So this is rather encouraging. And on this, thank you for your listening, and we'll be very happy to take any question you may have.
Thank you, Frederic, for your wonderful session here. You have linked about the anaerobic digestion and the importance of nanoparticles in enhancing the activity. And we are having some questions in the chat. Hello, can you hear me? Yeah. I hope in the real field applications, chances are very less for the nanoparticles to leach out. Do you have any experience where the analysis has been done in real wastewater treatment plants using the engineered nanoparticles?
On real wastewater treatment plants, there is some monitoring going on, but spiking, no, I don't think there have been any experiment or monitor. There is monitoring going on wastewater treatment plants, but not as we have done and the comparison with the metal salt. Also, you have to bear in mind, if you start to spike the wastewater, a real wastewater treatment plant, you might have some challenge for discharge at the end.
Okay. And can you give some insights on the possibilities of the problems associated with the agglomeration of engineered nanoparticles in the real-time sludge or process?
Well, can you repeat the question?
Yeah. What are all the possibilities of agglomeration of engineered nanoparticles in real-time sludge?
Yeah, it's very likely it will happen. Because effectively, as you could see from the slide, the nano are aggregating with the solids. So depending on your volume sludge index or your solid contents, the aggregation might be more pronounced in one wastewater treatment plant than another. So that is something you need to be aware, and also is something that at that time when we were doing analysis with the nanoparticles, is not as metal. So you can't, you need a much more cleaning step, and the ICPMS to do the analysis is more, require more pre-treatment to ensure you measure the salt or the metal, the nanoform. So at that time, we had challenge because it's very difficult to differentiate what is the nanoform and what is the metal salt form. The only way we could do it is because we had our control sludge, where what we measure we knew was the metal salt, and then we spike this sludge with the nanoparticles. So all excess or higher concentration was due to the presence of the nanoparticles. So we could subtract what we measure from the ENP spike minus the control. And that is not easy to quantify these in real sample.
Yeah. And is there any climatic or seasonal variation in regard to the effects of nanoparticles on wastewater treatment plants?
Any effect on what?
Seasonal variations or climatic variations.
Variations.
Yeah.
No, as far as I'm aware, no, but you might have, as any issues with wastewater, you got this diurnal. So you got the day and evening events where during the day, people are more at work and less taking showers, etc. So you might have more happening in the morning or in the evening rather during the day. And these are also weekends could be when you do your washing. So that could be a contribution to loads in the wastewater treatment plant.
Frederic, I think this question can be answered with respect to nano photocatalysis, right? Photocatalysis may have some specific climatic variations, no?
Yes, but here we are more, well, here you're more looking at source from product care or textile. So you're not in the photocatalysis. The nano will tend to be much more attached to the solids going to your wastewater treatment plant. So you slightly a different aspects for the topic we're talking today.
Yeah, and can you give some insights on the impact of immobilized microbes in activated sludge process, where you employ nanoparticles too?
Well, here we just show for the activated sludge is a biological process. So it relies on presence of microbes. And as you can see from the presentation, cell wall can be damaged, but we find also sometimes it was contributing to create more biofilms. So the nanoparticle are not always toxic, and there's homogeneous effects, so where it might stimulate or encourage activities. So the nano is not as straightforward as the metal, because it's not, they can have different effects, and it's all about dosing. So sometimes they have a positive effect, or sometimes might have a negative effect.
Yeah. And I think a couple of questions more. Did your study highlights anything about the effect of coliform bacteria in the treatment plants?
We did measure the coliform, but we didn't, as such, we didn't look at whether it reduced the pathogenic load or coliform loads in the treatment plants. We just look at microbial abundance. So we haven't specifically targeted like E. coli or any coliform. So we didn't look at log reduction over time, whether that was affected. And certainly something worth to look into. Our prime interest was to understand whether the biological process was affected.
Okay. I think, and how far the cost-effective strategies in engineered nanoparticles when you are scaling up the process, cost analysis, can you give some points?
Yeah, it could be expensive. You can source out engineered nanoparticles from suppliers, but effectively you wouldn't, it's the So I don't know in India or other kind of place, but as part of our projects, it was a good chunk of money being involved on getting the nanoform for our process. I can't remember off the top of my head, but I think it was in a few thousand to 10,000 pounds for the pilot line, because we've been running them for over 280 days. So you have to spike on a daily basis.
Okay, yeah.
To simulate inputs.
Yeah, that's right. And final question from my side, Frederic, from your presentation. You have given that the specific oxygen uptake rate increases in the metal salts on par with the nanoparticles. Whether you have tried with the different sizes of nanoparticles, whether it have some influence on your process?
Yeah, so in some instances, so the specific oxygen uptake rates, sometimes you see going up, and that is just indicating there is a stimuli effect due to a stress or so due to the presence of the metal. So the bugs are responding. And with the nano, sometimes we couldn't see much change, or a lower concentration, we find actually there were higher SUR response. So again, it's all about triggering stimuli or response to bugs. And with the metal, it's more, if you want clear, you got a clear cutoff, where the nano is not always, because you're going for high concentration, you stimulate or you inhibit. It could be lower concentration. The other point, which I haven't presented here, but here today was you got the mix together, silver, titanium, and zinc oxide. We did also some separate work where we look at individual metals, because when you got mixture, that do affect response as well.
Okay. And I think if we can we use some enzyme cofactors, metal cofactors for the hydrolyzing enzymes for the design of the nanoparticles, whether it will enhance the efficiency?
Could be. It's not something we've been looking at.
Okay.
Could be, yeah.
And thank you, Frederic. The final question from Paul. Can the treatments be conducted in triplicates to justify the process?
Again, costs and material you need to, so that is the big issues, but I think you're always on you're on the safe side, because here somehow you got three lines. One is the control, one is the spike with the metal, and the third one, the spike with the nano. So it's the same sludge, but you spike in with the metal salt or the nanoform, and you're trying to refer them. They operate in the same way. The only difference is spike. It would be nice to have all these triplicates, but you know, you are running pilot scale, so it's very hard to get these things replicated. So you have to compare between them, and the only things you could do is making your measurement in triplicates and comparing and doing them, doing your sampling or any change in the process, it should occur the same day for the three lines. So the work then you can't compare like for like. That is the main challenge when you are on a pilot scale. It would be nice to do everything in duplicates, because you have to bear in mind you need all the pumps as well all along. It's quite a lot of technical aspects to run along.
Yeah, thank you, Frederic, for answering the questions. Really, it is an informative session for the young researchers so that they can incorporate the nanoparticles based studies into their anaerobic digestion part. Thank you once again for your time, and thank you again.
Thank you to you for the invitation, and hope it was useful, and yeah, we're happy to contribute to this initiative. So thank you again for your invitation and to everyone attending today.
Yeah. Thank you, Frederic. Thank you. Thank you so much. Dear participants, kindly fill the feedback form so that you can receive the certificates. The certificates will reach you within 48 hours. Thank you.